about amino acids. We're going to finish this amino acid topic. Okay. Okay, so we're done with the introduction. Okay, and then a little bit of the structure of amino acid. How does the basic structure of amino acid? And then we have gone through the classification of amino acids and then characteristic. Either it is hydrophilic or hydrophobic or some kind like a hydrocarbon. And then we have learned about acid-base properties and then a little bit of the reaction. What happened to the amino acid? What happened to the amino acid if it is in the pH, in the certain pH? Okay, and then we have uncommon amino acid and non-protein amino acid. So we are going to make it a little bit faster for this part. Okay. Okay, we have. Okay, for all the details here, I need you just check out by yourself. Okay, it won't be too different. Just okay. What do we have here? Okay, so for the basic amino acid, last time last 2 days we have discussed about arginine. We have guanidinium, and then imidazole. The histidine we have imidazolium ring. Okay, and then for lysine we have the protonated alkyl amino group. Okay, so what is the use of the imidazolium ring? Later we will discuss during your enzymology next semester. Okay, we can purify this. We can purify the histidine. His tag protein. Okay, using the imidazolium. Okay. So yeah, for arginine and lysine, the side chain is fully. They are fully protonated at pH 7. Okay. Same either it is fully protonated or deprotonated. We need to go back to the principle. If the pH is higher than the PI, and then the ionizable group become deprotonated. If the pH is lower than its PI, the ionizable group become protonated. Okay. So what else? This one we have discussed. Okay, so we go to common amino acid in protein. Okay, we have not the 20 amino acid, they are common amino acid, but some uncommon amino acid, they, what happened? Okay, so arise by post-translational modification of protein, meaning that once the protein being translated, someone please turn off your mic. Okay, so please, okay, so if the protein, after the translation, okay, then you have the translated protein. Okay, so post-translational modification, meaning that the modification happened to the protein, okay, either glycosylation or disulfide bridge formation, or you can even create more ion pairs and everything. So the uncommon amino acid basically present after the protein is after the proteins are translated. Okay. So reverse modification, especially phosphorylation, and important integration is signaling. Okay. So modification, there are many reasons why do we need all this modification, but there are some of them are reversible. Okay. Okay, so we have amino acid less common, okay, found in certain protein like hydroxylysine, hydroxyproline. Okay, these two amino acids are found in collagen and gelatin protein. Okay, so hydroxylysine basically is from lysine, hydroxyproline basically from proline. Okay, and then yeah, a few in the alpha gamma carboxyglutamic acid found in several protein, in blood clotting, especially in snake, I think. Yeah. So these are basically this is the example. These are the example of uncommon amino acid in protein. For example, here, the principle, if that the principle will be the same. They should have the basic backbone, which is NCCN for the next amino acid, right? For the peptide bond. Okay, for example, here we have amino acid, chiral carbon, and then carboxyl group, carbon from carboxyl group. Okay, so except for proline, rather we call it as normal amino acid. We call it sometimes it's imino. Okay, so because the R group basically attached to the backbone. Okay, so for the tyrosine, still we have amino group, carboxyl, chiral carbon, and then carboxyl group. Okay, this will be the structure of the example of uncommon amino acid in protein. Okay, so for some of them, amino acid, they are not considered as protein. Okay, but the importance of having this non-protein amino acid is, you know, we cannot just simply, they are not say that they are not important. So basically they are important. Okay, so for example, we have here GABA and then histamine. Okay, and then we have adrenaline here, penicillamine, okay, and then a few more. Same, they're still amino acid, but they are not encoded for protein, okay? Still, they are still having amino group, chiral carbon, and carboxyl group. Amino group, chiral carbon, and carboxyl group. You see, if you look at the R group, they are really different than the one that we have in 20 common amino acid. Okay, so yeah, acid base properties we have covered, and then this also, what happened to this? Oh, this is yesterday's. Okay. Reaction of amino acid. Okay, you can have a look at this. Yeah, we are done with the basic with the amino acid topic. We're going to move to the second topic, which is protein. Okay, so in protein, okay, protein, the building block of protein, of course, from the amino acid. So now, why is only 9 minutes left? You have started earlier or what? Okay, so we have here protein. Let me just put it somewhere here. Okay, so more than 50% of dry weight of cell. Okay, we have unbranched polymers, amino acid link, head to tail. As usual, head to tail meaning that from the N terminal to the tail, which is C terminal. The repeat sequence will be NCC. Nitrogen from the amino group, chiral carbon, carbon from the chiral carbon, and then carboxy from the carboxyl group. So we can see here the repeat NCC, NCC, and all those things. Okay, carboxyl, oxygen, amide group are trans to each other. Okay, peptide bond, acid base. Okay, this one is nothing. Okay, so here what we want to check here about the torsion angle. So amino acid, if you notice one of the pictures from your friend, your super senior SCL, some of them, they did hang the amino acid, right? The model, right? Because basically for those who choose to hang the model, actually they do understand, because amino acid or protein, especially protein or peptide, they are not attached to something. They are just freely present in the cell, right? If it is in the beaker, if you extract the protein, meaning that the protein will not attach to the beaker or container. The protein will just float around the water. It will just mix up with the water. Okay, so the reason why they hang, basically they just want to show that, okay, the protein basically they are really flexible in the cell. Okay, so to make sure, we know that protein it will it will have the tertiary structure, right? Meaning that the 3D structure or 3D structure from the tertiary and quaternary structure, they make the protein become really complex. If it is in the primary structure or secondary structure, basically they cannot be functioning. Okay, so in order to protein, to be in 3D shape, of course it is determined, the 3D shape is determined by the weak molecular forces that we have discussed earlier. Okay, but they are also because of the torsion angle present in the amino acid, in the protein itself. Okay, for example, if you have the torsion meaning that the twist between the amino acid. Okay, so if we have the torsion angle between, the rotation between nitrogen from the amino group and then chiral carbon, which is nitrogen for the amino group and the chiral carbon. Hopefully you can see my mouse. Okay, amino group from the nitrogen, and nitrogen from the amino group, chiral carbon from the carbon from the chiral carbon. This torsion, the rotation here, we call it pi. Okay, if we had the torsion between chiral carbon and the carbon from the carboxyl group, this is called psi. Okay, maybe we got confused here. Carbon and carbon, chiral carbon, and the carbon from the carboxyl group, we call it psi. S psi. Okay, maybe you can just remember it as same. Okay, similar or sama. Okay, so for the chiral carbon with this nitrogen from the amino group, it is pi. If it is psi, another one will be pi. Okay, the torsion angle. Okay, so we will go in detail for this side. Okay, so you need to imagine that your protein basically is in this planar shape. In this plane. Okay, in this plane, you can the protein can be rotated. The amino acid can be rotated. Oops. Where is it? Okay, the torsion you can just twist between from here to this side or here to go to the other side. So the torsion between chiral carbon and the amino group and nitrogen from the amino group, this is pi, right? Okay, so the torsion angle, either you can make it positive or negative. Clockwise or anti-clockwise, something like that. How many degrees? Like 90 degrees clockwise or 180 degrees. So it will be the other way. Okay. So yeah, the rotation of this, the torsion between the because once the backbone is moved, okay, once the backbone is moved, of course the whole R group. Okay, can you imagine? We have the R group here, the hair here, very long hair here. Once the chiral carbon and the amino group move, then okay, the whole thing, the whole bunch of hair will be moved also. So if you have a really bulky R group, huge R group, okay, so the torsion basically will help you to maintain the structure. Maybe the structure itself, okay, for example, if it is too packed between this and this, maybe the structure will choose to use the other way around. Use the opposite side, something like that. Okay, we will see once we have got the model later on. Okay, so okay, another one here, we learn about the naming. Okay, we have the peptides here. If we have 2 amino acids, we call it dipeptide, 3 tripeptide, 4 tetrapeptide, pentapeptide, hexapeptide, heptapeptide. I believe we will be having hexapeptide and heptapeptide for your SDL, right? Okay, so if the peptide is longer, than 12, but less than 20 amino acid, we call it oligopeptide. Okay, your peptide, your amino acid or peptide chain longer than 12. Okay, but less than 20, we call it oligopeptide. Because bigger than 20, we can call mini protein. Okay, small size of protein. Okay, and then after that, of course, we call protein. Okay, so polypeptide, when the exceed several dozen, 100 up to 100 amino acid, and the protein, okay, protein. This protein, we have monomeric and multimeric, meaning that if monomeric, that means we have only one chain of protein. If we have multiple chain, we call it multimeric. Okay, if you have similar copies of the chain, we call it homomultimeric. If different copies of the monomer, we call it heteromultimeric. Okay, we are going to stop here since they're going to be less than a minute. We will continue later. We go with the second link, hopefully. Okay, see you. So I repeat, if you have one chain of the oligopeptide, or protein, okay, more than 100 amino acid, and then it is called monomeric protein if you have only one single chain. But if you have more than one, like 2, then we call it maybe dimer, okay, or trimer, or even tetramer. Even we have octamer protein. So octameric protein, meaning that we have 8 similar chain of protein, similar chain of the primary structure, okay, primary, amino acid repeat, okay, made up one big protein, okay, first chain, second chain, third chain, fourth chain, okay, until the 8, okay. So if we have similar protein, similar chain, we call it homomultimeric. But if we have, for example, tetra, 4 chain, right? 2 with, first one with the chain 1, chain A, chain B, chain C, or chain D. That means one protein having tetrameric, meaning that 4 chain, but different chain, okay? We call it heteromultimeric because they are having 4 different chain, okay? And then we have discussed on Monday about the size of protein. We measure the size of protein using Dalton. Like DNA, we measure the DNA based on the base pair, right? Between A and T, G and C, how many of these pairs, then we call it base pair. Okay, so for protein, the weight of the protein, the unit for the weight of the protein, okay, the molecular mass is called Daltons. And then how to write this? Oh my God, am I sharing? Sorry, I'm not sharing to myself. Okay, is it sharing now? Okay.
Yeah.
Okay, so molecular weight, we call it Dalton. Okay, so how to write it, just like on Monday, it will be D, capital D and A. This is Dalton. Okay, so if we have more than 1,000, then we add KDA. Okay, if we have more than 1,000 Dalton, then we can put small k, capital D, and A. Okay. So yeah, this one, just now we have discussed monomeric protein composition, monomeric, oligomeric, multi-subunit. Okay. Simple protein, peptones, peptide, albumin. Okay, conjugate protein. Okay, they have, okay, some protein, they have the prosthetic group. Okay, non-amino acid portion as certain molecule. For example, hemoglobin. The protein is globin, but the prosthetic group will be the heme group. Okay. How to, okay, I'm going to erase this. Okay. Okay, so for globular protein, okay, there are many types of structural protein. We have the globular protein and then fibrous protein. Okay, for the globular protein, we have the polar and charged group. For the fibrous protein, we have non-polar R group. Okay, so remember polar and charged group. Okay, we will have from the acidic and basic. And then for non-polar, we have from all the hydrocarbon stuff, the orange group. Okay, so this is the example. For example, we have insulin from bovine. Okay, the molecular mass, the weight of the protein will be 5,733 daltons. Is equivalent to 5.7 kilodalton. For cytochrome C from human, which is 13,000. That means 13 kilodalton. Ribonuclease A, 13.7 kilodalton. Lysozyme, which is egg white, okay, 13.9. It's almost similar to all these three. Okay, if you look at the numbers of amino acid residue, the bigger the protein size, the molecular weight, because they have the number of the amino acid residues are also big. For example, here glutamate dehydrogenase from bovine liver. The size is 1 million daltons, 1,000 kilodalton. Okay, because the amino acid itself is 8,300 amino acid residue. Okay, for your case, you need to have a model only 7 amino acid residue, really small. Okay, so even the number subunit, they are different. Subunits here, the either it is multimeric or monomeric just now, right? Okay, so insulin, even though it is small, like 51 amino acid, but it has 2 subunits. One is alpha, one will be beta. So the number of subunit will be different. Okay, alpha is different length, beta is different length, but the total number of amino acid is 51. Okay, so this one will be dimer. Okay, for insulin it is dimer and multimeric. Heterodimer. Okay, it is a combination between 2 different amino acid chain. Okay, for cytochrome C from human, it is bigger than insulin, but it has only 1 type of subunit. Okay, myoglobin 1, pancreas. Bovine chymotrypsin from bovine pancreas, it has 3. Okay, glutamate dehydrogenase, you see, it has 40 subunit. Okay, either it is different or not, either it is monomeric or hetero or homo. We can see in detail for this one. Okay. Okay, so this protein, okay, basically we can purify the protein. Okay, this technique, protein purification, is a technique where we can clean up the protein. We can polish the protein. Okay, there are a few techniques used to purify the protein. Purification means we want to polish the protein. Okay, for example, in your mouth itself, okay, you will have saliva, right? So in this saliva, you will find an enzyme, you will find, of course, water and other maybe microbes and everything in your mouth. Okay, so if you want to study about the enzyme in your mouth, which is amylase, right, where you start to digest your starch in, when you eat rice later, amylase will start to do its job first. Okay, so now if you want to study amylase in your mouth or in any other sources, so you need to separate this amylase from other impurities. Okay, the process of separating the protein of interest or enzyme of interest from the crude. Crude meaning that the mixture, the original source, is called purification. Okay, there are a few techniques to purify the protein. We will learn about protein purification during your enzymology class later. Okay, so this is example how it going to be looks like if you use the chromatography technique. Okay, so in this tube we call it column. Okay, glass column or even plastic, we have it. And then we have the stationary phase here. That means it is a bit inside, like a filter, something like that, inside the column. Okay, so once we have this the glass column, the column, together with the bead, the stationary phase here, the filter there, then this filter will start to differentiate what will be the first fraction. The fraction collector down here, tube number 1 until tube number 6 here. Basically, we have more, not only 6. We can even get hundreds of fractions. And then we start to investigate which fraction, which test tube will have the amylase that we want just now. Okay, just imagine your mouth. Basically, we cannot see, right, which one will be your amylase, which part will be only water or other contaminants. We don't know. So that's why we have a technique called protein separation or chromatography here. Then we can separate this protein. Okay, so maybe the first, for example here, 1 and 2 is just a water. And then 3 and 4 start, you can see the green color of protein coming out. And then maybe 5 and 6 until how many copies of the tube, then you can get whatever protein that you want. For example, in this case, maybe your protein of interest, the red color. So you need to wait from which fraction you can get the red color. Okay, so yeah. And then buffer, of course, will be used to just make sure your protein move. Like the buffer here represent the mobile phase, because the bead here will be the stationary phase, where the bead will capture your protein of interest. Okay, so yeah, once you have got your protein of interest, how do we check? Okay, this is called gel, polyacrylamide gel. Okay, you can use native page or SDS page. Okay, we will learn about the gel in the next section. But the gel is used to separate your protein of interest based on the molecular weight, the mass of the protein. Okay, MW represent the molecular weight. Okay, the bigger the protein, if your protein is big, just like cytochrome just now, right? Yeah. So it will appear at the top of the gel, because in principle you will load your protein up here. Okay, you will start to upload your protein here. This one we load with marker. Okay, and then the second, how do I move it? Okay, the second lane we load with our protein of interest. Okay, now if we have this marker, molecular weight, okay, marker meaning that the standard, like a ruler. Okay, the first top lane, the size will be 110 kilodalton. The second lane will be 90 kilodalton. Third lane here, 70 kilodalton. Fourth will be 50 kilodalton, 30 kilodalton, and then 10 kilodalton at the bottom. So when the electrical charge is applied to the gel, and then the current will move from the negative to the positive electrode. Okay, so what happened? Once you have loaded the protein here, it will start to move. Once the current is applied, okay, it will start to move until it will just, okay, I'm done. Okay, we need to apply the current until the separation of the marker. Until the markers are well separated. Okay, so initially the marker, of course, they're just packed together. But once we have run the gel, it will start to move slowly. Okay, the smaller protein, the smaller marker will move faster compared to the big one. Okay, if you have your protein of interest here, if you have your protein of interest, if you have your POI here, protein of interest here, and then you want to check, oh my God, my P is so bad, my I, oops. Never mind. It is POI. Okay, so once you start to apply the current, you can see that your protein of interest will move together with the marker, and then it will stop once it's reached this destination. Once you have stopped, because you found the lowest protein has reached the bottom of the gel, and then you will stop. Then you can see what is the size of your protein of interest. Okay, for this case, your protein of interest stop at 90, similar to this band, similar to 90 kilodalton band. That means your protein of interest size is 90 kilodalton. If your protein of interest stop somewhere in here, what do you think the size will be? Okay, you can type in the chat room.
40,000.
Oh my God, I don't know what happened to my 40,000. Okay, 40,000. Okay, so yeah, you got it right. That means how to stop this? Stop this. Okay, so that means if the protein of interest stop here, it is between 30 and 50. Then it will be 40 kilodalton. Okay, so before you start doing the purification, you need to know the size of your protein. Otherwise, what are you investigating? What are you checking? I'm checking your protein. What is the size of your protein? I don't know. And then how do you know your protein is there or not? I don't know. And then how can we help? Logically, you need to know the size of your protein of interest before you start running the gel. Otherwise, you don't know what size of protein are you checking, right? Okay, so if you don't know the size of your protein yet, but you do have the amino acid sequence, okay, don't worry, we have the ProgParam prediction tool, the SPC. So the marker and POI put the same time. Yeah, marker and the protein of interest, everything will need to be loaded at the same time. Okay, so we have basically this lane, this well, we call it well, okay, where we load the protein. This well, it will have 10 wells. So you can run the first well with a marker, second well with your protein of interest, the third well with your crude protein, a mixture of protein, unpurified protein. Okay, the fourth one may be the control or whatever you want to run. So because you will have 10 wells. Okay. Isn't the molecular weight experiment? Okay, the molecular weight of your protein of interest will not change. It will just be like that, okay, according to the marker that you have here. If you overrun, okay, you may be like you accidentally forget to turn off the current, the volt and everything. It like you just overrun it, maybe the fifth lane, I mean the 1, 2, 3, 4, 4. Okay, the 50,000 lane become at the bottom here. Okay, maybe here, just be here because you forget to turn it off. What happened? You will not see this band. You will no longer observe this band because the lowest part is already 50 kilodalton here, right? If the lowest part is 50 kilodalton and then your protein of interest, of course, will be exceeded here. I mean, your protein of interest, maybe it just turned out here. I mean, you know, it's gone, overrun. Okay, so hopefully I answer you. Okay, so what's next? Okay, what happened? Yeah, you can see many proteins later on here. Okay, so how can I, next one? Okay, so if you have like this different shape, I mean different size of, how do I erase this? Okay, if you have this different size of the protein, okay, then this size easily can be observed. What happened? This one, okay. You can see the different, different position of the band if you run all of them together, right? The glutamate dehydrogenase, of course, it will be at the top of the gel. But for this case, once we have the 1000 kilodalton, meaning that that gel just now is not suitable to be used. Okay, because the top marker only 110 kilodalton. Okay, we need to change the composition of the gel according to the size of the protein that we want to observe. Even this, starting from 145 until here, okay, we cannot observe using the 12% gel. We need to adjust the percentage of the gel. Okay, so meaning that the gel, okay, how can, okay, the gel, okay, the gel name just now, okay, you can have a look, even you can Google it to know more about the gel. It is called SDS page, okay, or native page, okay, and native page. Okay. SDS page stand for sodium dodacyl sulfate. Sodium dodacyl. Oops, where's the mouse? Where's the mouse? Okay, I lost my mouse again. Okay, sodium dodacyl sulfate polyacrylamide gel electrophoresis. So for native page, it's just a native original gel with polyacrylamide gel electrophoresis. Okay, please have a look. What is the difference between these two gels? When can we use these two gels? Okay. So, yeah, I think we can stop here first because we will go in detail when we have 2 hours maybe. Okay, so now we still have about a few minutes. Okay, any question you can just write down in the chat room, I mean chat box, chatting box. Stop sharing. Are you guys okay?
Yes.
Okay, so today we just cover about the uncommon amino acid and common amino acid, and then after that we stop. I mean, we're done with the amino acid, okay, and then we move to the protein, starting from primary structure. Primary structure we're done during the amino acid. This is a little bit of the protein structure, maybe secondary structure and quaternary structure. Okay. So what happened? We have learned about the psi and pi torsion angle. Okay. So, and then we learn about how to check the protein. Okay, the molecular weight of the protein. Okay, so maybe I can show you example of the gel. Okay, image. No, I don't want the recipe. I want the gel. Okay, so I'm going to share my screen again. Okay, so this is how it looks like. Okay, the gel will be like this. Okay, we have the molecular weight up here, the biggest one with 270 kilodalton, 250 kilodalton, 100 kilodalton, 85 kilodalton, 50, and the lowest one will be 17. Okay, the marker here represent the ladder here represent the marker. What is the size for the respective position? Okay, so now we can see here they are studying, I don't know which protein they are looking at. Okay, so they are studying the MHC protein and also the actin. So initially, before they start doing this, they know what is the size of MHC and then what is the size of actin. So if the actin is around, look at the picture, we know that the actin may be roughly around 45 kilodalton or 48 kilodalton, something like that, because it's closer to the 50 kilodalton. But why for MHC, maybe it's around 200 kilodalton. You can see here the thick band here around 250. This is the 250. This one is much lower than 250. Okay, we can see here the different treatment they have put. So without calcium chloride, 0 hour until 120 hours. You can see once you loaded the same amount of protein, you can see the different size of different intensity of the band. Okay, more intense of the gel, of the band here, the protein band here, meaning that the protein at that position is a lot. Okay, if it is very less, okay, for example here, with calcium chloride, the concentration here, the amount here, not concentration, the amount of protein here is much lesser than the first lane, right? The second lane here. Okay, the band will be the same size, but the amount of protein here, like 120, the last lane is lesser than the second lane. Okay, so how do we run this? Okay, so if you look at here, the way it is run, you can see here we have the buffer here, and then the buffer here with the glycine, tris glycine buffer. Okay, and then you start to run, you start to apply the electric after that. Okay, first all of the protein, they are all loaded inside the well. Okay, after you have loaded the protein into the well, then you apply the electric current, okay, electrical current, and then after some time, okay, the buffer will help to to regulate the positive and negative charge. It will pull the protein. Okay, so after that, once it is done, how do you know it is done? Because the ladder, they are all separated. Okay, what's next? Basically, once it is running, you don't know either you have completed or not, because the protein, even your marker, will be in the shape of colorless. There's no color. It's just transparent. Okay, only the dye here. Once you apply your protein sample here, you will put the dye, the blue dye. with the SDS and normally with the beta makeup to ethanol or something. And then, yeah, I can't remember the dye name. Okay, so the dye there will help you to it will help us, will guide us to stop. Okay, we need to stop running now because the dye is reaching the bottom. If you stop earlier, the protein marker or even your protein will not be able to separate well. So that's why you need to stop at the right time. Okay. Yeah, after you have done with the gel, we can start to stain with the Comasi blue staining, and then even we have the silver staining. Okay, so Comasi blue art normally we use is used to stain the protein. Okay, it is good to know what does Comasi blue art 250 or G250 stain. Okay, what in the protein being stained? Which part of the protein will be stained with this Comasi dye? Okay, we go back and imagine we have the backbone of the protein, NCCN, CCN, right? N-terminal carbon carboxy group, and the peptide bond, N-terminal carbon carboxy group, N-terminal, okay. And then also we have the functional group, right? The R group. And then which part of this backbone of your protein being stained with the Comasi blue staining? Okay, so basically we have one topic only for the gel during your analysis of biomolecule and enzymology class. We are not going to go too detailed with the gel, but it will be really interesting to know how to run the gel. Okay, so this is the tank, how it looks like, and then put it into the current, and then after that you can see here. And yeah, this is what happened. Yeah, everything is in the tank. You need to carefully load it, and then after that you can see different size of your protein. Okay, so yeah, any more question? Doctor, those SDS and NADPH are a technique for protein purification. Okay. So SDS or I would say electrophoresis technique. The gel. Gel for protein, right? Polyacrylamide gel electrophoresis is used to check the purity of the protein before and after the purification has been conducted. For example, initially, the protein, your protein, the crude protein. Which one will be the best one? Maybe initially, your crude protein meaning that the impure protein, the protein which contain all the impurities. Where's the gel just now? Okay, maybe for this one, initially, this is how it looks like. Okay, when we have here GST and fat lysate, meaning that this is the crude protein, the original protein. Okay, maybe this is your saliva. The first time you speak, hat-tree, make go. Okay, the first time you collect all the spit, your saliva. Okay, then when you run it into the gel, then you can see, wow, lots of other things. So happy to mix up with many things. Okay, and then second step, you may need to purify the protein. Remember, we have the column, we have the stationary phase. When we have the fraction collector there, it will start to track what will be the protein. What will be the protein in each of the collection, in each of the fraction. So maybe the fraction number 1 in the picture is nothing because it's just a buffer. Same goes to the fraction number 2. Maybe the third fraction, you start to observe the protein. Okay, maybe this is, this is marker, right? This is the one after the first step. In the third fraction. Okay, the third fraction will look like this. Majority, some of them have been removed from the gel. Okay, initially it is like this. It was like this, and then after that, okay, with the simple purification, a few steps, then you can see, okay, less impurities now. There's no more all these impurities down here, and then in middle of here, and then in between here, majority of them here has been removed. And then we check with the second lane. Okay, it reduced more. Okay, it is reduced. And then until the last lane, which is, it's not considered the last lane, in the fraction that when you want it, you can see, wow, the impurity, only a little bit of impurities there, but majority is the protein inside the fraction is coming from the protein of interest. So for this case, I would say, if we need to assess this gel, I would say we have managed to purify 90% of the protein of interest, the saliva just now, and the amylase just now. Fraction collected, the bit size is same to the tube or let the, okay. So, like I told you earlier, in protein purification, we have many steps, many different principles. Some of the protein, not some, protein can be separated based on size or molecular weight. Protein can also be separated based on the PI. Remember, we have the PI where the protein becomes zero net charge. Okay. Yes. Protein can also be separated based on the charge. If you have used pH 5 and the PI of the protein is pH 7, in pH 5, your protein basically will be protonated, right? So now the protonated protein, the protonated target protein can be purified based on the strength of the protonation. How many percent of the positively charged or negatively charged there. Okay, that means the protein can be separated based on the charge. And then protein can also be separated based on the hydrophobicity. Okay, and then also protein can be separated based on the affinity. Is there any special about the protein? Can we track the protein based on the tag? Based on that signal, something like that. Okay, so, but once we have made the protein become pure, then you can see from the gel. Wow, it's almost single band. I thought my spit is really clean. No, it's full with rubbish. Only after that, wow, I have a very pure amylase in my saliva now, for example. Okay. For the fraction collector, the bit size, okay, I have answered. Is it, is it to check the purity before and after purification? Yes. This is used to check the quality of your purification. Okay, it's going to end at any time. So I'm going to stop sharing. If you have any other question, you can just type in the Discord channel. Basically, right after the class, if you still remember what kind of question you want to ask, you can just go to Discord and start typing there. If you think that question is really general, can help other people to understand too, just type in the general channel. Okay. Don't feel like, wow, am I going to ask the most stupid question? No. Basically, you actually help other people who doesn't know what to ask. The problem with the student, you don't know what you don't know, right? That's normal. You don't know what you know. You don't know what you don't know. Yeah, me too. I don't know what I know. Yeah, that's the thing. Okay, so just ask anything you want. Okay, so yeah, if you think about that thing may help your friend, just ask how to cast the gel, what happened to the gel. Okay, it's going to end at any time. So see you guys. I didn't manage to see all of your faces. Are you there?